How does your body decide how much to eat? Why does skipping just one nutrient have such, well, dramatic effects? And what's really happening inside when you gain or lose weight?
Yeah, these aren't simple questions at all. They really get to the absolute core of our physiology, this incredibly complex dance between the energy we take in, the energy we use up, and all those vital nutrients that make it all possible.
And that's exactly what we're diving into today. We've got this fantastic stack of material provided by you, our listener, and it lays out these fundamental concepts, energy metabolism, regulation, vitamins, minerals, in some pretty serious detail.
hmm Our mission here really is to guide you through this specific source material. We want to pull out the most important nuggets of knowledge, connect the dots for you, and help you build a level of understanding that honestly feels like mastery of these complex systems, the kind of grasp professionals in the field work with.
Right. Think of this as our joint expedition into this knowledge base. I'm bringing the curiosity and maybe pointing out the fascinating bits on the map.
And I'm here to provide the expert synthesis, helping us navigate the terrain, understand the significance, and really see how all these pieces fit together.
Okay, let's jump right in then. At its most basic level, the whole story of body weight and composition starts with a pretty simple concept: balance.
Precisely. It's the first principle laid out in this material. The stability of your body weight, your composition over time, it's a direct reflection of balancing the energy you get from food against the energy your body expends.
Yeah.
Basic functions, activity, all of it. It sounds simple, but there's just so much complexity underneath.
And when that balance gets thrown off, you see pretty predictable results, right?
Absolutely. If your energy intake consistently exceeds your energy expenditure, if you're overfed, physiologically speaking, that excess energy gets converted and stored mainly as fat.
Uh-huh.
And this leads to an increase in body weight and, over time, potentially to obesity.
And the flip side, not enough energy coming in.
Well, insufficient intake, where your expenditure is higher than your intake, forces your body to start using its own mass for fuel. That means a loss of body weight, and if it's severe and goes on long enough, it can lead to starvation.
But this material quickly points out it's not just about the total calories, is it? Like, you can't just run on pure sugar or something.
That's a really crucial point. Energy balance determines weight, yeah, but maintaining actual health requires the right balance among proteins, carbohydrates, fats, and ensuring you get all the necessary minerals and vitamins too. Every single metabolic system in your body needs specific building blocks and cofactors to work properly, not just the raw energy units.
Okay, speaking of raw energy, the source gives us some very specific numbers for the energy content of the macronutrients when they're fully oxidized in the body.
Yes, these are foundational numbers to keep in mind. A gram of carbohydrate, when it's fully oxidized, gives you 4.1 calories.
Oh.
A gram of fat yields a pretty significant 9.3 calories.
Wow.
And an average gram of protein gives about 4.35 calories.
So fat clearly packs the most energy per gram. But the material also brings up this idea of physiologically available energy. What's that distinction about?
Right, so that number accounts for the fact that our digestive system isn't like 100% efficient.
Yeah.
We don't absorb every single bit of energy from the food we eat. Some is lost.
Okay.
So for carbohydrates, about 98% gets absorbed. That makes its physiologically available energy closer to the familiar 4 calories per gram.
Right.
For fat, absorption is typically around 95%, which gives us that commonly cited figure of 9 calories per gram.
Mm-hmm.
And for protein, about 92% is absorbed, which also works out to about 4 calories per gram physiologically available.
So those are the familiar 494 numbers we often see. That 9 for fat really stands out compared to carbs or protein, doesn't it?
It really does, and that has major implications for how easily different foods can contribute to an energy surplus and, storage.
This material also shows the pretty striking difference in macronutrient makeup across different diets. Like, a typical American diet gets about 15% of its energy from protein, 40% from fat, and 45% from carbohydrate.
Yeah, and you contrast that with diets in many non-Western countries, where carbohydrates often provide the vast majority of energy. Fat and protein combined might only be, say, 15 to 20% of total calories. It really highlights not just cultural food differences, but how different food structures deliver energy.
And looking at examples like those in Table 72-1 really makes it concrete. Meats, obviously high in fat and protein,
Like bacon or beef in the examples.
while grains and vegetables are clearly the carb powerhouses. Corn, bread, things like that.
Exactly.
The source also makes this interesting point about how deceptive fat can be because of its density.
Oh, this is key. Fat, in its pure form, is often nearly 100% fat by weight, But proteins and carbohydrates, they're usually found in a watery medium. They often make up less than 25% of the total weight of the food itself. Think about that butter pat versus potato example the material uses. A small pat of butter can easily contain as much energy as a large helping of potato, just because the butter is so much more concentrated energy by weight. It's a really vivid way to understand how energy density impacts calorie intake.
Right, that makes sense. Okay, let's move beyond just the energy provision and dive deeper into protein's essential roles. The source really emphasizes it's far more than just a calorie source.
Oh, absolutely. The average daily requirement is stated as 30 to 50 grams. But the reason we need this daily intake is fascinating.
It's because our body is constantly breaking down its own protein stores, isn't it?
Exactly. Every single day, around 20 to 30 grams of our body's own proteins get degraded. These amino acids then get reused for other things, sure, but the body needs a constant supply of dietary protein to rebuild. To resynthesize the proteins, it's just broken down. It's this continuous cycle of breakdown and repair.
And not all dietary protein is created equal in this process. The source talks about partial proteins. What does that mean?
Yes, so this refers to proteins that don't have adequate quantities of one or more of the essential amino acids.
Okay, and essential means?
Essential means our bodies can't make them themselves. We absolutely must get them from our food. So if a protein source is low in even one of these essential amino acids, the body just can't efficiently use that protein source to replace the body proteins. It's degraded. It's like trying to build something with missing parts.
And this is why animal proteins are generally seen as more complete.
Generally, yes. They tend to have a better balance of all the essential amino acids we need. Vegetable and grain proteins, on the other hand, often have what are called limiting amino acids, meaning one or two essential amino acids are present, but in insufficient amounts to fully support protein synthesis on their own.
The source uses specific examples, like corn being low in tryptophan and lysine.
Right, and legumes, like beans and lentils, tend to be inadequate in methionine.
This is a really cool insight from the material. These incomplete proteins can actually complement each other.
Precisely. This is the principle of protein complementation. By combining different plant-based protein sources, say eating corn and legumes together, or even just within the same day, you can get all the essential amino acids you need. The combination provides a complete protein profile, overcoming the limitations of each food individually. It's a really key concept for understanding nutritional adequacy, especially in plant-based diets.
And what happens if someone's diet relies too heavily on just one incomplete protein source? Like, the material mentions diets where cornmeal is the main protein.
Yeah, that can unfortunately lead to a severe protein deficiency syndrome called kwashiorkor. The source lists the classic symptoms: failure to grow, profound lethargy, sort of depressed mentality, and generalized edema that's swelling caused by fluid retention.
And that edema, the swelling, is directly linked to the lack of protein.
Yes, absolutely. When you don't get enough protein, your body struggles to make important plasma proteins, like albumin, in the liver. These proteins are crucial for maintaining the osmotic pressure in your blood, keeping fluid inside the blood vessels. When albumin levels drop too low because of protein deficiency, fluid leaks out of the capillaries into the spaces between tissues, causing that characteristic swelling or edema. It's a powerful illustration of how vital protein is, even for something as fundamental as fluid balance.
The source also introduces this idea of protein sparing. What's that about?
This is a key metabolic concept. Basically, when your diet gives you plenty of carbohydrates and fats, your body prefers to use those for energy. They effectively spare the protein from being broken down just for fuel.
Oh.
In that situation, most of your energy comes from the carbs and fats, and the protein you eat is reserved for its more critical roles, building tissues, making enzymes, antibodies, that sort of thing.
But the opposite happens in starvation, doesn't it?
Tragically, yes. Once your carbohydrate stores are gone and your fat stores start getting seriously depleted, the body has no choice but to turn to its own protein for energy. The material states that during prolonged starvation, body protein can be consumed very rapidly, sometimes at rates of several hundred grams per day.
Wow, compared to the normal 30, 50 grams turnover, that's huge.
It's a massive difference. It really highlights how quickly the body starts breaking down its essential structures, its functional tissues. When fuel is critically scarce, the priority becomes generating energy, even at the cost of losing vital mass.
All right, let's shift gears slightly. How can we actually measure what fuel the body is burning at any given time? This material introduces the concept of the respiratory quotient, or RQ.
The RQ is a really elegant physiological tool. It's defined simply as the ratio of the volume of carbon dioxide your body produces to the volume of oxygen it consumes. So CO2 output divided by O2 usage over a specific time period.
And measuring this gives a snapshot of metabolism.
Exactly. By measuring this ratio, you get a real-time glimpse into the fuel mix your cells are actually metabolizing at that moment.
And the RQs for different macronutrients, carbs, fats, proteins, are distinct because of their chemical makeup, right?
Precisely. Their chemical structures dictate how much oxygen is needed and how much CO2 is produced when they're burned. For carbohydrates, the RQ is exactly 1.0.
Why 1.0?
Because when you completely oxidize a simple carbohydrate molecule, like glucose, the reaction uses 1 molecule of oxygen for every molecule of carbon dioxide produced. O2 in equals CO2 out, ratio is 1.
Makes sense.
For fat, however, the average RQ is significantly lower, around 0.70.
Okay.
And for protein, it's intermediate, roughly 0.80.
So why are the RQs for fat and protein lower than for carbs? What's going on chemically?
Right, it comes down to the hydrogen content. Fat and protein molecules contain proportionally more hydrogen atoms relative to oxygen compared to carbohydrates. When these molecules are metabolized, some of the oxygen you consume is used not just to form CO2, but also to combine with these excess hydrogen atoms to form water, H2O.
Ah, okay.
But less CO2 gets formed relative to the amount of oxygen used up, resulting in a lower CO2-O2 ratio, a lower RQ.
So just by measuring the gases someone breathes in and out, what the source calls the respiratory exchange ratio, you can estimate their body's average RQ at that time.
Exactly. If you measure that exchange ratio and it's close to 1.0, you know the person is primarily burning carbohydrates. If it's closer to 0.70, they're mainly metabolizing fats. An intermediate RQ, say 0.85, suggests a mix of carb and fat burning. And as the material points out, if you also estimate protein metabolism separately, which we'll get to, you can calculate the fuel mix with even greater precision.
And what kind of key metabolic insights can we get from looking at RQ changes based on the source material?
Oh, the shifts in RQ are really telling about the body's state. For instance, immediately after you eat a mixed meal, your RQ will typically approach 1.0.
Because you're burning the incoming carbs first.
Exactly. The body prioritizes using those readily available carbohydrates. But then, maybe 8 to 10 hours after that meal, once those readily available carbs are used up, the RQ will drift down towards 0.70.
Signaling the switch to fat burning.
Right. The body is now relying more on stored fats for energy. And a really striking insight comes from looking at untreated diabetes mellitus. In that condition, the RQ tends to stay down near 0.70 even after eating carbohydrates.
Why is that?
Because without adequate insulin action, the body's cells can't effectively take up and use glucose from the blood. So even though carbs are available, the cells are forced to rely primarily on fat metabolism for energy. The RQ reflects this inability to utilize carbohydrates properly.
It's amazing how much that simple ratio can reveal about what's happening inside. Now, you mentioned estimating protein metabolism separately using nitrogen excretion. How does that work?
Right, so this method leverages the fact that protein is unique among the macronutrients because it contains nitrogen, about 16% nitrogen by weight on average.
Okay.
When protein is broken down in the body, this nitrogen has to be dealt with and is eventually excreted. About 90% of this excreted nitrogen leaves the body through the urine, mainly in the form of urea, but also some uric acid and creatinine. The other 10% or so is typically lost in the feces.
So you measure the nitrogen in the urine?
Exactly. You typically collect urine over a full 24-hour period and measure the total nitrogen content. Then you estimate the amount lost in feces by adding about 10% to the urinary nitrogen value. This gives you an estimate of the total nitrogen lost from protein breakdown over that day.
And then you multiply that by 6.25. Where does that number come from?
Good question. Since protein is about 16% nitrogen, that means 1 gram of nitrogen comes from roughly 100 divided by 16, which equals 6.25 grams of protein.
Ah, okay, it's the conversion factor.
Right, so you multiply the total estimated daily nitrogen excretion, urine plus 10%, by 6.25, and that gives you a pretty good estimate of the total amount of body protein that was broken down during that 24-hour period. The source gives an example. If someone excretes 8 grams of nitrogen in their urine per day, you estimate total excretion is 8 plus 0.8 equals 8.8 grams. Multiply by 6.25 and you get about 55 grams of protein breakdown.
And this allows you to track something called nitrogen balance, right?
Precisely. You compare the nitrogen intake from the protein you eat with the nitrogen excretion from protein breakdown. If your intake is less than your excretion, you're in negative nitrogen balance.
Meaning you're losing body protein.
Exactly. Your body is breaking down more protein than it's taking in and synthesizing, so your overall protein stores are decreasing. This is a really critical marker of nutritional status, especially in situations like illness, injury, or starvation. Positive nitrogen balance, where intake exceeds excretion, occurs during growth, pregnancy, or recovery from illness.
Okay, understanding how the body uses fuel and how we measure that leads us straight into maybe the most complex part of this material. How the body actually regulates all this food intake, energy storage. It's clearly not just about whether food is available or not.
Oh, far from it.
Hmm.
Maintaining a stable and adequate energy supply is absolutely fundamental for survival. Because of this, the body has evolved these incredibly powerful, often redundant physiological control systems. They govern how much we eat, how much energy we expend, and how much energy we store away.
The source makes a distinction between short-term and long-term regulation. What's the key difference there?
It really comes down to purpose and timescale. Short-term regulation is mainly focused on preventing you from overeating during a single meal. It's about controlling the size of the meal you're consuming right now.
Okay.
Long-term regulation, on the other hand, is geared towards maintaining the body's overall energy stores, primarily fat stores, at a relatively constant level over much longer periods, like days, weeks, and years. It's about maintaining that overall energy balance.
Let's quickly clarify some key terms related to eating that the source uses: hunger, appetite, and satiety.
Right, these are important. Hunger is that primal physiological drive to eat. It's often accompanied by physical sensations, like stomach contractions, and it motivates the search for food.
Okay.
Appetite's a bit different. It's more of a desire for food, often for a specific type of food. It's influenced more by learned preferences, habits, sensory cues like smell or sight, and psychological factors. It guides food selection.
Yeah, satiety.
Satiety is the feeling of fullness and satisfaction that occurs after eating. It's the signal that inhibits further eating, basically telling you the meal is over. These sensations, hunger, appetite, satiety, they're all modulated by this incredibly complex interplay of physiological signals from the body, plus environmental cues, cultural factors, and our own learned behaviors.
And the central command and control for a lot of this seems to be located in the brain, specifically in the hypothalamus.
The hypothalamus is definitely highlighted in this material as a primary orchestrator of food intake control. It contains several distinct clusters of neurons, or nuclei, that seem to have opposing functions in regulating feeding.
Like the lateral nuclei.
Yes, the lateral nuclei of the hypothalamus are often referred to as the feeding center. If you stimulate this area in an animal experimentally, it causes voracious eating hyperphagia.
Wow.
Conversely, if you destroy these lateral nuclei, the animal loses all desire for food, leading to severe weight loss and a state called inanition. It seems these nuclei are involved in exciting the motor drives that make you actively seek out and consume food.
And then there are the ventromedial nuclei.
Right. These are generally considered the major satiety centers. Stimulating these nuclei causes complete satiety. An animal will refuse even the most tempting food, a state called aphagia.
So the opposite effect.
Exactly. And if you destroy the ventromedial nuclei, the result is relentless, voracious eating that continues even when the animal is full, leading to extreme obesity. In some experiments mentioned, animals' body weights quadrupled. So these nuclei seem to provide that crucial inhibitory signal, the sense of nutritional satisfaction that suppresses the feeding center.
The source mentions other hypothalamic nuclei involved too, not just those two.
That's right. The paraventricular nuclei are mentioned lesions there can also lead to excessive eating. The dorsomedial nuclei lesions there can depress eating. And critically, the arcuate nuclei are identified as a major integration point. They sit in a key location to receive hormonal and nutrient signals from the body.
So the hypothalamus isn't working in isolation. It's constantly getting input from all over the body.
Absolutely. This material details the huge variety of signals that feed into the hypothalamus, as shown in figures 72-1. You've got neural signals coming up from the GI tract, especially strep signals from the stomach, indicating fullness, which travel via the vagus nerve.
Okay.
Chemical signals directly from nutrients circulating in the blood. The brain senses levels of glucose, amino acids, and fatty acids, and these can influence satiety.
Mm-hmm.
Hormonal signals released from the GI tract itself in response to food passing through. Hormones released from adipose tissue, like fat cells, providing feedback about the status of long-term energy stores. And then there are higher-level inputs from the cerebral cortex, related to things like the sight, smell, and taste of food, which strongly influence our appetite and eating behavior.
It really is a major information processing hub. And the source notes there's a high concentration of receptors in the hypothalamus for all sorts of substances that either stimulate feeding or inhibit it.
Right. These signaling molecules are broadly categorized as either orexigenic, meaning appetite stimulating, or anorexigenic, meaning appetite inhibiting. Table 72.2 lists a whole bunch of them, just showing how complex the chemical signaling involved is.
Within those arcuate nuclei you mentioned, the source highlights two specific key populations of neurons that act as central controllers. Figure 72-2 illustrates this really well.
Yes, these are absolutely critical. The POMC neurons and the NPY-AGRP neurons. POMC neurons produce a large precursor molecule called pro-opiomelanocortin, or POMC. This gets chopped up into several active peptides, most importantly alpha-melanocyte-stimulating hormone, or ADO-MSH. These neurons also produce another peptide called CART.
And what do they do?
When these POMC neurons are activated, they decrease food intake and simultaneously increase energy expenditure. They're essentially the body's stop eating, burn more fuel command center.
Okay, the brakes. And the other group?
The other group are the NPY-AGRP neurons. These produce neuropeptide Y, NPY, and a GUYTON-related protein, AGRP. Activation of these neurons has the exact opposite effect. They powerfully increase food intake and reduce energy expenditure. They're the eat more, conserve fuel command center.
So they're like these opposing forces constantly influencing our drive to eat and how much energy we burn.
Exactly. And they don't just act independently, they influence each other, and they are major targets for many of the hormonal signals we talked about. For instance, hormones like leptin from fat cells, insulin from the pancreas, and CCK from the gut tend to activate the POMC neurons and inhibit the NPY-AGRP neurons, thus reducing appetite. Ghrelin, on the other hand, the hunger hormone from the stomach, primarily activates the NPY-AGRP neurons, stimulating hunger. So the arcuate nuclei really serves as this crucial integration point, receiving signals related to both immediate meal intake and the status of long-term energy stores, and then adjusting the output of these opposing neuronal systems.
This material puts a lot of emphasis on the system that stems from those POMC neurons, the melanocortin system. It links it really strongly to regulating energy stores and even to severe obesity when it's defective.
Yes, this system is incredibly powerful and important. The MSH released from the POMC neurons acts on specific receptors called melanocortin receptors, particularly types MCR3 and MCR4. These receptors are freely concentrated in other brain areas involved in energy balance, like the paraventricular nuclei.
Okay, and what happens when MSH activates these MCR receptors?
Activating MCR3 and especially MCR4 has a strong anorexigenic effect. It significantly reduces food intake, but it also increases energy expenditure.
How does it increase energy expenditure?
The source notes that activating MCR4, particularly in the paraventricular nuclei, stimulates pathways that lead down to the brainstem, specifically the nucleus tractus solitarius, NTS. This, in turn, increases the activity of the sympathetic nervous system, which ramps up metabolic rate and heat production.
So this pathway acts as a major brake on appetite and simultaneously presses the gas pedal on metabolism.
That's a good way to think about it. And its importance is really underscored by genetic studies. The material states that mutations in the gene for POMC itself, or in the gene for the MCR4 receptor, are the most common single-gene causes of human obesity identified so far. They might account for something like 5 to 6% of cases of severe early-onset obesity.
Wow. So if this system isn't working properly, the brake is essentially off, and maybe the metabolic rate is suppressed too.
Exactly. The body isn't getting that crucial stop eating, burn energy signal effectively.
And on the flip side, can excessive activation of this melanocortin system suppress appetite too much?
Yes, that's hypothesized to be a factor in the severe anorexia, the loss of appetite, that's often seen in conditions like severe infections, cancer, or chronic kidney failure, uremia. The inflammatory processes in these diseases might overactivate this powerful appetite-suppressing pathway.
Okay, what about AGRP, that peptide released by the opposing NPY-AGRP neurons? What does it do?
AGRP, a GUYTON-related protein, acts as a natural antagonist, a blocker, for the MCR3 and MCR4 receptor. It basically binds to those same receptors that MSH activates, but instead of activating them, it blocks MSH from binding and having its effect.
So it cancels out the stop eating signal.
Precisely. By inhibiting the effects of MSH, AGRP promotes feeding. While its exact day-to-day role is still being fully worked out, the source notes that excessive production of AGRP, sometimes due to genetic variations, is also associated with increased food intake and obesity.
And NTY, the other peptide from those neurons.
NPY, neuropeptide Y, is another extremely potent orexigenic peptide. Its release is strongly stimulated when the body senses that energy stores are low. NPY powerfully stimulates appetite, driving feeding behavior. And importantly, it also acts to inhibit the activity of the POMC neurons, further tipping the balance toward increasing food intake and conserving energy. It's a major signal saying, we need fuel.
Beyond the hypothalamus, the material mentions other brain areas involved in the whole process of feeding.
Right. It makes a distinction that the brainstem, lower down, contains the centers that control the mechanical act of feeding. The coordination of salivation, licking, chewing, swallowing.
Uh-huh.
Experiments mentioned show that these basic motor patterns of feeding can still occur, even if the brain is surgically disconnected above the brainstem but below the hypothalamus. So the hypothalamus and higher centers aren't controlling the mechanics per se, but they provide the drive, the motivation, and control the quantity of food consumed. They excite these brainstem centers to carry out the action.
And higher centers like the amygdala and parts of the prefrontal cortex also contribute maybe more to the qualitative aspects of appetite.
Yes, they're highly interconnected with the hypothalamus and influence appetite control. The amygdala, which is heavily involved in processing emotions and sensory information, especially smell, olfaction, has areas that can both increase and inhibit feeding. Stimulation in some amygdala regions can even trigger mechanical feeding behaviors.
The material makes an intriguing point about what happens if the amygdala is damaged.
Yes, it mentions that destruction of certain parts of the amygdala can lead to a condition called psychic blindness, specifically regarding food choice. The animal or person might lose the ability to select food based on quality, preference, or past experience. They lose the usual appetite control that governs what type of food they seek out, not just how much. It highlights how these higher centers add complexity to appetite beyond just basic hunger and satiety.
Okay, let's bring this back to how these regulatory systems work in practice. Looking first at the short-term signals that help us stop eating at the end of a meal.
Right, the signals that induce satiety during a meal. The primary one mentioned is simple physical feedback. Gastrointestinal filling.
Just the stomach stretching.
Exactly. As your stomach and the upper part of your small intestine, the duodenum, distend with food, stretch receptors embedded in their walls get activated. They send signals up to the brain, primarily via the vagus nerve, which then act to inhibit those feeding centers in the hypothalamus. It's a basic negative feedback loop saying, okay, we're getting full, helping to limit the size of that single meal.
And then there are several hormones released from the gut during digestion that also signal satiety, right?
Absolutely. A number of gut hormones play a role here. CCK, or cholecystokinin, is a key one. It's released from the duodenum and jejunum, parts of the small intestine, mainly in response to the presence of fats and proteins entering from the stomach.
And how does CCK signal satiety?
It seems to act locally, activating sensory nerve endings in the gut wall. These signals then travel up the vagus nerve to the brain, contributing to the feeling of fullness and satiation during the meal. The source notes, however, that CCK's effect is relatively short-lived. It's important for helping to end the current meal, but probably doesn't have a major impact on regulating energy balance over the long term.
Okay. Then there's PYY.
Yes, peptide YY. This hormone is secreted by cells throughout the GI tract, especially further down in the ileum and colon. Its release increases after food intake, particularly after meals rich in fat, and levels peak about 1-2 hours later.
And its effect.
Animal studies suggest PYY can decrease food intake for several hours after a meal. Its precise contribution to regulating appetite in humans is still being actively researched, but it's considered another important gut-derived satiety signal.
GLP-1 is another one mentioned, often linked to satiety and also insulin.
Right. Glucagon, like peptide 1, GLP-1, is released from intestinal cells when food, especially carbohydrates and fats, enters the intestines. It's well known for enhancing glucose-dependent insulin secretion from the pancreas, helping manage blood sugar after a meal. But both GLP-1 itself and the resulting rise in insulin levels also tend to suppress appetite, signaling back to the brain. So the act of digesting food triggers this hormonal cascade that promotes feelings of fullness.
And what about the hormone that seems to signal hunger before a meal starts?
That would be ghrelin. It's unique among the gut hormones discussed here because it stimulates feeding. It's orexigenic. Ghrelin is released mainly by specialized cells, oxyntic cells, in the stomach lining.
And its levels change with meals.
Dramatically. Ghrelin levels rise significantly during periods of fasting, peaking just before you typically eat a meal, and then they fall rapidly after you eat. This pattern strongly suggests it acts as a key physiological signal helping to initiate feeding, basically telling your brain time to eat.
The source also mentions something about the metering of food by oral receptors. What does that refer to?
Ah, yes. This refers to the sensory input you get just from the act of food passing through your mouth, the chewing, salivation, tasting. Even before food hits your stomach and triggers those stretch and hormonal signals, these oral sensations can provide some short-term inhibition of the feeding center.
But it's not as strong a signal.
No, the material indicates this effect is less potent and much shorter-lived than the signals from GI filling or hormones. Maybe it lasts only 20 to 40 minutes. It's thought to contribute to satiety, but it's not sufficient on its own to regulate intake accurately over time. Think of it as an initial quick assessment, while the gut signals provide more sustained feedback. These short-term signals collectively work to optimize the size of each meal and match it to digestive capacity.
So while those short-term signals are managing the meal itself, it's really the intermediate and long-term regulatory systems that try to keep our overall energy stores stable over time.
Exactly. These longer-term systems are tuned more to the body's overall nutritional status, the amount of energy stored away. Although it's crucial to remember, as the source points out, that in humans, these biological controls are heavily influenced, sometimes overridden by behavioral choices, social contexts, cultural norms, and the surrounding food environment.
The classic theories about long-term regulation relate to the brain sensing nutrient levels in the blood, right? The static theories.
Yes, the glucostatic, aminostatic, and lipostatic theories. They're based on the idea that the brain, particularly the hypothalamus, continuously monitors the blood concentrations of key nutrients. Glucose from carbohydrates, amino acids from protein, and certain metabolites derived from fat breakdown, like keto acids and specific fatty acids.
And changes in these levels trigger hunger or satiety.
That's the core idea. Decreased levels of these circulating nutrients are thought to signal a state of energy deficit, leading to the sensation of hunger and driving feeding behavior aimed at restoring normal blood concentrations and replenishing body stores. Conversely, elevated levels after a meal contribute to satiety.
And this links back to those specific hypothalamic neurons we talked about earlier.
Precisely. Experimental evidence supports this. For example, rising blood glucose levels have been shown to increase the electrical firing rate of neurons in the satiety centers, like the ventromedial and paraventricular nuclei, while simultaneously decreasing the firing rate in the feeding center, the lateral hypothalamus. Amino acids and lipid metabolites appear to have similar, though perhaps less potent or direct, effects on the activity of these or closely associated neuronal populations.
Temperature regulation also seems to interact with feeding control.
It does. The source notes that exposure to cold temperatures tends to increase food intake, while exposure to heat generally decreases intake. This interaction happens within the hypothalamus, linking the systems that regulate body temperature and energy balance.
And that makes adaptive sense, right?
Absolutely. Eating more in the cold provides the extra fuel needed to increase metabolic rate and generate more body heat. It also provides substrate for potentially building up insulating fat stores. Conversely, reducing intake in the heat helps minimize the metabolic heat produced from digesting and processing food.
But perhaps the most powerful long-term signal discussed in this material, especially regarding fat stores, seems to be leptin.
Yes, leptin is highlighted as a really key hormonal signal originating directly from adipose tissue from your fat cells. As the amount of fat stored in your body increases, your adipocytes produce and release more leptin into the bloodstream.
So it's a signal proportional to fat mass. Where does leptin go and what does it do?
Leptin travels through the bloodstream to the brain. It's able to cross the blood-brain barrier and bind to specific leptin receptors located in several critical brain areas involved in energy balance. These include those key arcuate nuclei, both the POMC and NPY-AGRP neurons, the paraventricular nuclei, the ventromedial nuclei, and even areas in the brainstem.
And what's the fundamental message leptin is delivering to the brain?
Leptin is essentially the body's fat stores communicating with the brain, signaling that there are sufficient or even excessive energy reserves stored away. Its binding to those receptors triggers a whole cascade of downstream actions, primarily aimed at decreasing further fat storage.
How does it do that? What are the specific actions?
Leptin does several things simultaneously. First, it inhibits the activity of those appetite-stimulating NPY-AGRP neurons in the arcuate nucleus, reducing the production of NPY and AGRP. Second, It activates the appetite-inhibiting POMC neurons, increasing the release of AMSH, which then acts on melanocortin receptors to suppress appetite and boost energy expenditure. Third, it increases the production and release of other anorexigenic substances, like corticotropin-releasing hormone, CRH, in the hypothalamus. Fourth, it increases sympathetic nervous system activity, which, as we discussed, leads to increased metabolic rate and energy expenditure, burning more calories. And fifth, it tends to decrease insulin secretion by the pancreas, which reduces the drive to store energy.
Wow, that's a really comprehensive system for basically saying, okay, we have enough energy stored, time to stop eating so much and burn more.
It really is. It acts as a powerful negative feedback signal from fat stores to the brain. And its importance is starkly illustrated by those rare but informative cases of congenital leptin deficiency or defective leptin receptors in humans. As the source mentions, these individuals experience intense hyperphagia, uncontrollable hunger, and develop morbid obesity very early in life, mirroring what's seen in animal models lacking leptin signaling.
This brings up a really critical question that the material addresses directly. If leptin is supposed to signal sufficiency and reduce appetite, why do most obese people actually have high levels of circulating leptin, not low levels?
Yes, this is a crucial paradox and a major focus of obesity research. The leading hypothesis, as discussed in the source, is the development of leptin resistance.
Meaning the brain stops listening to the signal. Essentially, yes. Despite having very high levels of leptin circulating in their blood, the brains of many obese individuals seem to become resistant or less sensitive to its effects. The leptin receptors themselves might become downregulated or desensitized, or the signaling pathways downstream from the receptor might become impaired. So the brain isn't adequately hearing or responding to the signal that indicates sufficient energy stores. The stop eating, burn more message isn't getting through effectively.
Or, as the source also suggests, maybe it's not just resistance, but also the influence of other redundant regulatory systems, or perhaps the powerful impact of our modern environment, the easy availability of palatable food, social factors, stress that can override even a strong leptin signal. It's almost certainly a complex interplay of factors. The precise molecular mechanisms of leptin resistance are still being actively investigated, but the general principle holds. The body has these long-term systems, like leptin signaling, designed to sense and regulate energy stores over time, working in concert with the short-term signals that manage individual meals, all aiming to maintain energy balance. The challenge arises when these systems face conditions they didn't necessarily evolve for, like our modern food environment.
Okay, this detailed look at regulation leads us directly into the topic of obesity itself, how it's defined, its prevalence, the underlying mechanisms, and its complex causes and consequences.
Right, so obesity is fundamentally defined as an excessive accumulation of body fat. Quantitatively, the source gives general cutoffs, typically considered over 25% of total body weight as fat in men and over 35% in women.
But the most common way it's assessed in practice is using BMI, right? Body mass index.
Yes, BMI is widely used as a practical surrogate marker because directly measuring body fat percentage is more complex. BMI is calculated simply as weight in kilograms divided by height in meters squared, kilograms.
And the categories.
Overweight is typically defined as a BMI between 25 and 29.9 kilograms of common starch. Obesity is defined as a BMI of 30 kilograms or greater.
But BMI has its limitations, doesn't it? Like, it doesn't distinguish between fat mass and muscle mass.
That's its main drawback, exactly. A very muscular athlete might have a high BMI that puts them in the overweight or even obese category, even though they have very low body fat. That's why the source emphasizes that for assessing health risks associated with excess weight, the distribution of fat is actually more critical than just the total amount.
Particularly the visceral fat.
Yes, visceral or abdominal adiposity, the fat stored deep within the abdominal cavity around the internal organs, is much more strongly linked to metabolic complications like insulin resistance, type 2 diabetes, fatty liver disease, hypertension, cardiovascular disease, and even certain cancers compared to subcutaneous fat, the fat stored just under the skin.
And there are simpler ways to get an idea of abdominal fat, like measuring waist circumference or the waist-to-hip ratio.
Exactly. The source provides typical cutoff points used in the U.S. A waist circumference greater than 102 centimeters, about 40 inches, for men, or 88 centimeters, about 35 inches, for women, or a waist-to-hip ratio greater than 0.9 for men or 0.85 for women are generally considered indicators of increased health risk due to abdominal obesity.
The prevalence numbers for obesity mentioned in the source are pretty staggering.
They really are. It's described as a major public health crisis, a global epidemic, particularly in industrialized nations. The source highlights the rapid increase, especially in the U.S., where prevalence has reportedly risen by over 30% in just the past decade. Current estimates suggest around 70% of U.S. adults are now classified as either overweight or obese, with over 35% falling into the obese category, BMI equals 30.
And at its absolute core, the mechanism is still that simple energy balance equation we started with, isn't it?
Fundamentally, yes. Obesity results from a sustained period where energy intake consistently exceeds energy expenditure. That excess energy has to go somewhere. And the body's primary way of storing large amounts of excess energy is as fat, triglycerides, in adipose tissue. The source reminds us of the energy density. For every 9.3 excess calories consumed that aren't burned off, about 1 gram of fat gets stored.
Where does this fat get stored primarily?
Mainly in subcutaneous adipose depots, under the skin, and within the abdominal cavity, visceral fat. But excess fat can also accumulate in other places that's not ideally meant to be, like the liver, leading to fatty liver disease, muscles, pancreas, and other tissues, which can contribute to metabolic dysfunction.
The material also provides some updated insights into how the fat cells themselves, the adipocytes, change during obesity. It's not just about existing cells getting bigger.
Right. The older view was perhaps simpler, that the total number of fat cells was largely set during childhood and adolescence, hyperplasia, and adult onset obesity was primarily due to those existing cells just getting larger by accumulating more fat, hypertrophy.
But newer research suggests.
Newer research, as detailed here, indicates that new fat cells can actually be formed from precursor cells, predipocytes, even in adulthood, especially in response to significant and prolonged positive energy balance.
So significant adult obesity often involves both an increase in the number and the size of fat cells.
Correct. The source states that obesity developing in adulthood is typically characterized by both hypertrophy, larger cells, and hyperplasia, more cells. It even gives a striking statistic. An extremely obese person might have up to 4 times the normal number of adipocytes, and each of those cells might contain up to twice the normal amount of lipid. It represents a massive expansion of the body's fat storage capacity.
And when someone reaches a stable, albeit obese, weight, it means their intake and expenditure have balanced out again, just at that higher level of fat storage.
Precisely. The body has found a new equilibrium, but it's an equilibrium characterized by excess fat mass. To lose that excess fat, the energy balance equation must be actively shifted back into negative territory. Expenditure must exceed intake.
Okay, let's talk causes. The source makes it clear that obesity is complex, resulting from an interplay of genes, lifestyle, and the environment.
Absolutely. While genetic factors undoubtedly play a role in determining an individual's susceptibility to gaining weight, the sheer speed and scale of the global obesity epidemic over recent decades strongly implicates major shifts in our lifestyle and environment as the primary drivers. Our genes simply haven't changed that rapidly.
A major culprit identified is the increase in sedentary lifestyles.
Unsurprisingly, yes. Lack of physical activity reduces energy expenditure, decreases muscle mass, which is metabolically active, and favors fat accumulation. The source directly links this to the rise in sedentary behavior, spending long hours sitting, increased screen time, less physically demanding jobs, and transportation.
And muscle activity is a big part of energy expenditure.
It's the single largest variable component of daily energy expenditure for most people. It might account for only 20–30% in a very sedentary person, but it can rise to 60–70% in someone doing heavy physical labor. So increasing physical activity is a powerful lever for increasing energy expenditure and helping to tip the balance towards weight loss or maintenance. The source also notes that strenuous exercise can even boost your resting metabolic rate for a period after the activity itself.
Abnormal feeding behavior, influenced by our modern environment, social norms, and psychological factors, is also highlighted as a major contributor.
Definitely. The environment in many industrialized nations presents what's sometimes called an obesogenic environment. This includes the widespread availability, accessibility, and affordability of highly palatable, energy-dense foods, often in large portion sizes, combined with those sedentary lifestyles. On top of that, psychological factors play a role for many individuals. Things like chronic stress, anxiety, depression, or using food to cope with emotions can lead to patterns of overeating or making less healthy food choices.
The source also points a finger at childhood overnutrition as a potentially critical factor setting the stage for lifelong obesity.
Yes, this is an important point. The period of infancy and childhood is when fat cells are rapidly forming and proliferating. Overfeeding during these critical developmental windows can lead to the establishment of a significantly larger number of adipocytes compared to a child with normal nutrition.
And these extra cells stick around?
Generally, yes. The total number of fat cells tends to remain relatively stable throughout adulthood, although individual cells can shrink or expand. So having a higher number of fat cells established early in life might predispose an individual to easier weight gain and make weight loss maintenance more challenging later on. The source cites this statistic that something like 80% of obese children go on to become obese adults, suggesting that early life factors can have a lasting impact.
Now, genetic factors definitely play a role too, right? Obesity clearly runs in families.
Oh, absolutely. Heritability estimates suggest that genetic factors contribute significantly, perhaps accounting for 20-25% or even more of the variation in susceptibility to obesity within a population.
How might genes influence obesity risk?
Genes can influence many aspects of energy balance. They can affect the structure and function of those appetite control centers in the hypothalamus we discussed, influence baseline metabolic rate or the tendency to fidget, non-exercise activity thermogenesis, affect how efficiently energy is stored as fat versus burned off, and influence pathways related to nutrient sensing and hormone signaling.
The source mentions some specific rare single gene causes of obesity again here.
Yes, it reiterates those monogenic forms where a mutation in a single gene has a very large effect, leading to severe obesity.
Yeah.
Mutations in the POMC gene or the MCR4 receptor gene are highlighted as the most common known monogenic causes, though still accounting for only maybe 5-6% of severe early onset cases. Congenital leptin deficiency or mutations in the leptin receptor are even rarer examples.
But for the vast majority of people with obesity, it's not just one faulty gene.
Correct. Most common forms of obesity are considered polygenic. This means that many different genes likely contribute small effects to overall susceptibility. An individual might inherit a particular combination of these gene variants that slightly increases their risk, but whether or not they actually become obese depends heavily on the interaction between this genetic predisposition and environmental factors like diet, physical activity, and other lifestyle choices.
The source then introduces another layer of complexity: epigenetic mechanisms, potentially connecting parental obesity to offspring risk across generations.
Yes, this is a fascinating and rapidly evolving area of research, illustrated in Figure 72-3. Epigenetics refers to changes in gene expression, which genes are turned on or off, that occur without altering the underlying DNA sequence itself. These changes can sometimes be influenced by environmental factors and potentially be inherited.
And the hypothesis here is
The hypothesis discussed is that environmental factors associated with parental obesity, perhaps the parent's diet, metabolic state, or exposure to certain stressors, could lead to epigenetic modifications in their sperm or egg cells. These modifications might alter the expression of genes involved in appetite regulation, metabolism, or fat storage in the developing fetus.
wow.
Furthermore, the intrauterine environment itself during pregnancy could induce epigenetic changes in the fetus. And potentially, epigenetic changes could even occur in the fetal germ cells, the precursors to sperm and eggs, raising the possibility of effects being transmitted to subsequent generations.
Is there strong evidence for this in humans yet?
The source notes that much of the supporting evidence currently comes from experimental animal models. While the extent to which these mechanisms contribute to human obesity is still under active investigation and requires more research, it's a compelling idea that adds another dimension to how obesity risk might be transmitted or amplified across generations.
But regardless of the precise contribution of epigenetics, the source points back to that dramatic rise in childhood obesity.
Yes, the fact that childhood obesity prevalence has more than tripled globally since the 1970s points unmistakably towards powerful environmental and lifestyle shifts as the primary drivers in recent times, interacting with existing genetic susceptibilities.
Finally, the source mentions that direct physical damage to the brain, particularly the hypothalamus, can cause obesity.
Yes, we touched on this earlier with the experimental lesions of the ventromedial nuclei causing hyperphagia. In humans, conditions like certain types of brain tumors, particularly hypophyseal tumors that press on or damage the nearby hypothalamus, can sometimes disrupt appetite regulation and lead to significant weight gain and obesity.
But what about obese individuals who don't have any obvious structural brain damage?
Right. The source speculates that there might be more subtle, functional abnormalities in the organization or signaling within the feeding centers of the hypothalamus in many obese individuals. Perhaps differences in the balance of neurotransmitter release, receptor sensitivity, or pathway connectivity.
And this idea is supported by observations about what happens after weight loss.
Yes, particularly the phenomenon often referred to as metabolic adaptation, or the set point theory. Many individuals who have been obese and then lose a significant amount of weight experience several challenges that make maintaining that weight loss incredibly difficult. One is persistent, often intense, hunger.
Uh-huh.
Another is that their resting metabolic rate often slows down more than would be predicted just based on the loss of body mass alone. It's as if the body is actively fighting to regain the lost weight, defending a higher set point for body fatness.
And this adaptation involves hormonal changes too.
Yes, the source details accompanying changes in circulating hormones that promote weight regain. After weight loss, levels of the hunger-stimulating hormone ghrelin tend to be increased, while levels of satiety hormones like leptin and PYY and appetite-suppressing signals like IMSH are often decreased. These hormonal shifts can persist for a year or even longer after weight loss, constantly signaling increased hunger and potentially reduced energy expenditure, which helps explain why sustained weight loss solely through dieting is so challenging for many people. The body's biology seems geared towards regaining that lost weight.
Okay, given this incredibly complex picture of causes and the body's resistance to change, how does the source material approach the treatment of obesity?
Well, the fundamental principle remains the same. To achieve weight loss, you have to create and sustain a negative energy balance. Energy intake must be consistently lower than energy expenditure to force the body to tap into and burn its stored fat reserves.
The NIH guidelines mentioned in the source offer some specific strategies for achieving that caloric deficit.
Yes, they provide targets. For individuals who are overweight or have moderate obesity, typically BMI 25-35, a daily caloric deficit of about 500 kilocalories below their maintenance needs is often recommended. Since 1 pound of fat represents roughly 3,500 kilocal, this deficit aims for a gradual weight loss of about 1 pound per week.
And for more severe obesity,
For individuals with a BMI over 35, a larger deficit of 500 to 1,000 kilocal per day might be suggested, targeting a weight loss of 1 to 2 pounds per week. The typical initial goal often cited is achieving a meaningful, but perhaps modest, 10% reduction in body weight within the first 6 months.
and the source makes a point about being careful about nutrient intake during calorie restriction.
Absolutely crucial. When you're significantly reducing overall food intake, it becomes even more important to ensure you're still getting adequate amounts of essential vitamins, minerals, and protein to prevent deficiencies. This requires careful food choices or sometimes supplementation.
But, as we just discussed, calorie restriction alone is often not enough for long-term success due to those adaptive responses.
Exactly. The source strongly emphasizes that increased physical activity combined with reduced caloric intake is essential for a successful long-term weight reduction and, perhaps even more importantly, weight maintenance. You really have to address both sides of the energy balance equation consistently. Lifestyle modification focusing on both diet and exercise remains the cornerstone of obesity management.
Pharmacological management using medications is also part of the treatment landscape, although the source suggests it has its challenges.
Yes, the material discusses several different types of drugs that have been used or are currently used for weight management. Some historically aim to suppress appetite by directly inhibiting those feeding centers in the hypothalamus, like amphetamines and related compounds. But these often came with significant side effects and abuse potential.
What about current options?
Sympathomimetic drugs, like phentermine, which is sometimes used in combination with topiramate, work partly by stimulating the sympathetic nervous system, which can reduce appetite and slightly increase metabolic rate. However, this stimulation carries the risk of overexciting the cardiovascular system, potentially increasing blood pressure and heart rate.
And the source notes that one such drug, sibutramine, was actually withdrawn from the market.
Yes, sibutramine was removed from the U.S. market back in 2010 due to findings of an increased risk of heart attack and stroke in patients using it. It serves as a clear example of the safety concerns that can arise with drugs affecting the sympathetic nervous system. Another drug mentioned, lorcaserin, which worked by activating specific serotonin receptors involved in satiety, was also withdrawn by the FDA in 2020 due to safety concerns flagged in post-market studies.
Are there newer drugs targeting different pathways, like those POMC neurons we talked about?
Yes, there are combination therapies like bupropion plus naltrexone, which are thought to work, at least in part, by increasing the activity of those appetite-inhibiting POMC neurons. Another important class mentioned are the synthetic GLP-1 receptor agonists. These drugs mimic the action of the natural gut hormone GLP-1, which promotes satiety and also enhances insulin secretion. They were initially developed for type 2 diabetes, but were found to cause modest but consistent weight loss, likely through effects on appetite centers in the brain, possibly involving the melanocortin pathway, as well as slowing stomach emptying.
And some drugs work differently, affecting nutrient absorption or excretion.
Right. The SGLT2 inhibitors, another class of drugs primarily used for type 2 diabetes, cause some weight loss by blocking the reabsorption of glucose and sodium water in the kidneys, leading to excess glucose being excreted in the urine. Then there's orlistat, which works locally in the gut. It inhibits pancreatic and gastric lipases, the enzymes needed to digest dietary fat. This reduces fat absorption, causing undigested fat to be excreted in the feces.
But orlistat is known for having some unpleasant gastrointestinal side effects and can affect the absorption of fat-soluble vitamins.
Correct. Those are common issues with orlistat.
Yeah.
So the overall summary provided in the source regarding pharmacological treatment is that currently approved long-term weight loss medications generally produce modest weight loss, on average, often in the range of 5% to 10% of initial body weight, sometimes a bit more for the newer agents. They tend to be most effective when used as part of a comprehensive program that also includes significant lifestyle changes, diet, and exercise. They are tools, but not magic bullets.
And for the most severe cases of obesity, surgery becomes a potential option.
Yes, bariatric surgery is typically considered for individuals with morbid obesity, usually defined as a BMI of 40 or greater, or for those with a BMI of 35 or greater who also have serious obesity-related health complications like uncontrolled type 2 diabetes, severe hypertension, or sleep apnea that haven't responded adequately to other treatments.
What are the main types of bariatric procedures described in this material?
The source mentions the most common ones. Gastric bypass, specifically Roux-en-Y gastric bypass, involves creating a very small stomach pouch and then directly connecting this pouch to the jejunum, a lower part of the small intestine, bypassing most of the stomach and the duodenum. This restricts intake and also alters gut hormone signaling. Gastric banding involves placing an adjustable silicone band around the upper part of the stomach, creating a small pouch above the band, which restricts how much food can be comfortably eaten at one time. And vertical sleeve gastrectomy involves surgically removing a large portion, about 80% of the stomach, leaving behind a narrow, tube-like or sleeve-shaped stomach remnant. This also restricts intake and affects gut hormones.
And these procedures generally lead to pretty substantial weight loss.
Yes. On average, bariatric surgery leads to significantly more weight loss than non-surgical approaches for severely obese individuals. And interestingly, the source notes that procedures like gastric bypass and sleeve gastrectomy often lead to a rapid improvement or even remission of type 2 diabetes and hypertension, sometimes occurring very quickly after surgery, even before substantial weight loss has happened. This suggests mechanisms beyond just weight loss, likely related to altered gut hormone profiles and nutrient flow.
But it's important to remember these are major surgeries.
Absolutely. The source appropriately emphasizes that these are major operations with potential risks and complications, both short-term and long-term. While they can be highly effective for weight loss and improving related conditions in carefully selected patients, the full long-term effects on overall health, mortality, and potential nutritional deficiencies are still being studied and require lifelong follow-up.
Okay, we've spent a lot of time on excess energy storage and obesity. Now let's look at the other extreme described in the material. Conditions involving unintended weight loss like inanition, anorexia, and cachexia.
Right. Inanition is basically defined as extreme weight loss, resulting from any cause. It's the physiological opposite of obesity. It could simply be due to inadequate food availability, starvation, but often, as the source discusses, it results from various pathophysiological conditions that decrease the desire or ability to eat.
Such as?
These could include psychogenic factors, like severe depression, dysfunction within the hypothalamic appetite centers, or signals released from diseased peripheral tissues that suppress appetite. An important point made is that inanition is often associated with an increased rate of energy expenditure relative to body size, which seems counterintuitive but contributes to the rapid weight loss.
The term anorexia in this physiological context has a specific meaning, doesn't it? It's not just referring to the eating disorder, anorexia nervosa.
Correct. The source defines anorexia more broadly here as a reduction in food intake caused primarily by a diminished appetite, emphasizing the role of central neural mechanisms that control hunger and satiety signals. This distinguishes it from simply not eating due to other factors like physical pain, nausea, difficulty swallowing, or lack of access to food. Anorexia nervosa is then described as a specific psychic state where there's a profound loss of desire for food, often accompanied by aversion or even nausea at the thought or sight of food, leading to severe self-induced inanition.
And cachexia. That sounds different again.
Cachexia is distinct and particularly devastating. It's defined as a complex metabolic disorder characterized by inflammation, insulin resistance, and an abnormally increased rate of energy expenditure and muscle protein breakdown. This leads to involuntary weight loss, particularly loss of muscle mass, that is often greater than what can be explained by reduced food intake alone.
So it's not just lack of appetite, the body is actively wasting away.
Exactly. Cachexia often occurs alongside anorexia. The combination is sometimes called the anorexia-cachexia syndrome, and it's tragically common in many chronic diseases. The source highlights its prevalence in over half of all cancer patients, in AIDS wasting syndrome, and in other chronic inflammatory conditions like severe heart failure or rheumatoid arthritis.
What mechanisms does the source propose for cancer-induced anorexia and cachexia specifically?
It's understood to be driven by a complex interplay between the tumor itself and the body's response to it, involving both central neural effects and peripheral factors. The source specifically points to the role of pro-inflammatory cytokines, signaling molecules like tumor necrosis factor alpha, TNS-DOME, interleukin-6, IL-6, and interleukin-1, IL-1, as well as other factors released by the tumor or the body's immune cells, like a proteolysis-inducing factor that directly promotes muscle breakdown.
And how might these cytokines cause the anorexia part?
The hypothesis presented is that these inflammatory cytokines act on the brain, particularly the hypothalamus, to cause anorexia, partly by activating that same hypothalamic melanocortin system we discussed earlier, the body's powerful brake on appetite.
So the inflammation is hijacking the appetite control system.
That seems to be part of the mechanism. The source mentions that animal studies have shown that blocking the hypothalamic melanocortin receptors can partially alleviate the anorexia and cachexia seen in some cancer models. However, it also emphasizes that the precise mechanisms are complex and likely involve multiple pathways, and developing effective therapies specifically targeting cachexia in humans remains a major challenge and area of ongoing research. It's a combination of reduced intake and accelerated, disordered energy expenditure and tissue breakdown.
When unintended weight loss is severe and prolonged, whatever the cause, it eventually leads to starvation, the systematic depletion of the body's stored energy reserves in a fairly predictable pattern.
Yes, the body prioritizes fuel sources as starvation progresses, as illustrated in Figure 72-4 from the source material.
Carbohydrates are the first to go, right?
Correct. The body's stores of readily available carbohydrates, primarily glycogen stored in the liver and muscles, are relatively small. We only have maybe a few hundred grams, enough to meet the body's energy needs for perhaps half a day or so under basal conditions. These stores are used up very quickly in the initial hours of fasting or starvation.
Then the body turns to its main long-term energy reserve,
which is fat. An average non-obese person has vastly more energy stored as fat, potentially 100 times more than as carbohydrate. Fat becomes the primary fuel source during sustained starvation. Its breakdown and utilization continue progressively, essentially unabated, until most of the fat stores are eventually exhausted.
And what about protein? How are protein stores utilized during starvation? It happens in phases.
Yes, the pattern of protein depletion described is more complex, occurring in roughly 3 phases.
Initially, there's a relatively rapid depletion of the most easily mobilized protein stores. These amino acids are used either directly for metabolic needs or, importantly, converted into glucose, gluconeogenesis, primarily to supply fuel for the brain, which normally relies heavily on glucose.
Okay.
Phase 2. After these readily available protein stores are used up, the rate of protein breakdown slows down significantly. The rate of gluconeogenesis from amino acids decreases perhaps to about 30–50% of its initial rate. This phase coincides with a major metabolic shift.
The shift towards using ketone bodies.
Exactly. As glucose availability decreases and fat breakdown ramps up, the liver starts producing significant amounts of ketone bodies, like beta-hydroxybutyrate and acetoacetate from fatty acids. A key adaptation during prolonged starvation is that the brain gradually adapts to using these ketone bodies as a major alternative fuel source. The source suggests they can eventually supply up to 2/3 of the brain's energy needs.
And this adaptation seems designed to protect the body's protein stores.
It absolutely is. By reducing the brain's demand for glucose, which would otherwise have to be made largely from protein, the increased use of ketone bodies helps to spare the body's remaining more critical protein structures during this intermediate phase of starvation. This protein-sparing effect is crucial for survival. However, this phase can only last as long as there are sufficient fat stores remaining to fuel ketone body production.
What happens in the final third phase of protein depletion?
Phase 3 begins when the body's fat stores become almost completely depleted. At this point, protein is essentially the only significant energy source left. The body is forced to turn back to breaking down its functional proteins at a rapid rate again.
And this leads to?
This rapid final depletion of essential proteins ultimately leads to death. Since proteins are critical for virtually all cellular functions, enzymes, transporters, structural components like muscle, immune function, vital physiological processes begin to fail as these critical protein structures are consumed for energy. The source states that death usually occurs when total body proteins have been depleted to about half their normal level.
The source also makes a crucial point about vitamin deficiencies becoming a serious compounding factor during starvation.
Absolutely. While energy depletion is the primary issue, the lack of incoming food also means a lack of essential vitamins. The body's stores of many vitamins, particularly the water-soluble B vitamins and vitamin C, are quite limited compared to fat stores.
So deficiencies can develop relatively quickly.
Yes, mild deficiency symptoms might start appearing after just a week or two of severely inadequate intake for some vitamins, becoming severe and potentially life-threatening after several weeks or months, depending on the vitamin and initial stores. These deficiencies don't just happen in isolation, they significantly worsen the overall state of debility caused by the energy deficit itself and definitely contribute to the eventual outcome.
The source gives some specific timelines.
It mentions, for example, that severe scurvy, vitamin C deficiency, might take 20 to 30 weeks to develop to the point of death, while symptoms related to deficiencies in most B vitamins, except B12, which has longer liver storage, can appear within days or weeks and become severe relatively quickly. These micronutrient deficiencies are an integral part of the pathophysiology of starvation.
That paints a very clear and honestly quite stark picture of how the body consumes itself when deprived of external fuel. Let's shift our focus now completely to those essential micronutrients themselves, starting with the vitamins.
Okay. Vitamins, as defined here, are essential organic compounds that the body needs, typically in very small quantities, to facilitate normal metabolic reactions. Crucially, our body cells generally cannot synthesize them, or not in sufficient amounts, so they must be obtained from the diet. Lacking a specific vitamin disrupts specific metabolic pathways, leading to characteristic deficiency syndromes.
And the amounts needed daily vary quite a bit, as shown in Table 72-3. Storage in the body varies too.
Yes, requirements differ significantly between vitamins. Storage also varies greatly. While all cells store tiny amounts, major storage depots exist for the fat-soluble vitamins, particularly vitamin A and vitamin D, mainly in the liver. These stores can sometimes last for months, even with deficient intake.
But water-soluble vitamins?
Most water-soluble vitamins, like the B-complex vitamins and vitamin C, have only very limited body storage capacity. This means reserves can be depleted relatively quickly, sometimes within days or weeks, if dietary intake becomes inadequate. Vitamin B12 is a notable exception among the water solubles, as the liver can store significant amounts that may last for years.
Okay, let's dive into some of the key vitamins detailed in this material, looking at their main function and the consequences of deficiency. First up, vitamin A.
Vitamin A exists in different forms, preformed retinol from animal sources, and provitamin A carotenoids, like beta-carotene, from plants, which our body can convert to retinol. The source highlights two primary functions. First, it's essential for forming the retinal pigments, like rhodopsin, in the eye's retina, which are absolutely critical for vision, especially in dim light, night vision. Second, vitamin A is crucial for the normal growth, differentiation, and maintenance of epithelial cells throughout the body, the cells lining our skin, respiratory tract, digestive tract, urinary tract, etc.
And what happens when you don't get enough vitamin A?
The earliest and most specific sign of deficiency is usually night blindness, due to insufficient retinal pigment regeneration. As deficiency becomes more severe, the problems with epithelial tissues become widespread. The source lists: the skin becomes dry, rough, and scaly, acne can develop, growth failure occurs in children, reproductive function is impaired, testicular atrophy in males, disrupted cycles in females.
And effects on the eye beyond night blindness?
Critically, yes. The epithelial lining of the cornea can undergo abnormal changes, becoming dry and keratinized, hardened. This condition, called xerophthalmia, can progress to corneal ulceration, scarring, and potentially irreversible blindness. It's a major cause of preventable blindness in children in some parts of the world.
And the link to infection?
Because vitamin A is so important for maintaining the integrity of those epithelial barriers, deficiency makes these surfaces much more vulnerable to infections. That's why vitamin A is sometimes referred to as the anti-infection vitamin.
Okay, let's move to thiamine, which is vitamin B1.
Thiamine's active form in the body is thiamine pyrophosphate, TPP. This acts as an essential coenzyme, sometimes called co-carboxylase. Its critical role is in the decarboxylation, removal of CO2, of pyruvic acid and other related alpha keto acids. This step is absolutely vital for channeling the products of carbohydrate breakdown, pyruvate, into the citric acid cycle for energy production. It's also needed for metabolizing certain amino acids.
So, central to energy metabolism, especially from carbs, and deficiency leads to the classic syndrome beriberi.
Yes, beriberi results from severe thiamine deficiency. Because thiamine is so crucial for carbohydrate metabolism, its deficiency impairs the ability of tissues, especially the nervous system and heart, which rely heavily on glucose, to extract energy efficiently.
What are the main features of beriberi described here?
The source describes major effects on the nervous system and the cardiovascular system. Neurologically, deficiency leads to lesions in both the central and peripheral nervous systems. This can manifest as decreased glucose utilization by the brain, nerve cell degeneration and demyelination, loss of the protective sheath around nerves. Clinically, this results in symptoms like painful polyneuritis, inflammation of multiple nerves, muscle weakness and atrophy, and potentially paralysis. Cardiovascularly, severe thiamine deficiency weakens the heart muscle itself, contributing to cardiac failure.
It also affects blood vessels.
Yes, a characteristic feature is significant peripheral vasodilation. The blood vessels in the limbs relax and widen. This reduces peripheral resistance and increases the amount of blood returning to the already weakened heart, leading to a state called high-output cardiac failure, often accompanied by significant edema, swelling, and fluid accumulation in the abdomen ascites.
And there are GI disturbances too.
Indigestion, severe constipation, loss of appetite, anorexia, reduced muscle tone in the stomach, gastric atony, and decreased secretion of stomach acid are common. These are thought to result from impaired function of the smooth muscle and glands in the GI tract due to the overall depression of carbohydrate metabolism needed for their function. The combination of these neurological, cardiovascular, and gastrointestinal symptoms constitutes the clinical picture of beriberi.
Next, riboflavin, vitamin B2.
Riboflavin is a component of two extremely important coenzymes, flavin mononucleotide, FMN, and flavin adenine dinucleotide, FAD. These act as crucial hydrogen carriers, electron carriers, within the mitochondrial oxidative phosphorylation system, the electron transport chain, where the vast majority of cellular ATP, energy currency, is generated.
So, also central to energy production.
Oh, well.
Severe deficiency is dramatic in animal studies.
Yes, the source notes that severe deficiency in experimental animals can cause profound effects like dermatitis, vomiting, diarrhea, muscle spasticity, eventually coma and death, reflecting a severe depression of fundamental oxidative processes.
What about in humans?
Severe deficiency is less common in humans, but mild to moderate deficiency is thought to be relatively common globally. Symptoms are often less dramatic and perhaps less specific than beriberi or pellagra. They can include digestive disturbances, a burning sensation of the skin and eyes, cracking at the corners of the mouth, a condition called angular stomatitis or chylosis, headaches, mental depression, and forgetfulness. Riboflavin deficiency often occurs alongside deficiencies of other B vitamins like thiamine and niacin, contributing to the complex picture of malnutrition syndromes.
Speaking of niacin, vitamin B3.
Niacin, which can also be synthesized in limited amounts from the amino acid tryptophan, is a critical component of two other major coenzymes, nicotinamide adenine dinucleotide, NAD, and nicotinamide adenine dinucleotide phosphate, NADP. These function as essential hydrogen acceptors and donors for a vast number of dehydrogenase enzymes involved in virtually all pathways of oxidative metabolism, breaking down carbohydrates, fats, and proteins to generate energy, ATP.
And its deficiency causes the disease pellagra.
Yes, pellagra is the classic severe niacin deficiency syndrome. The underlying problem is a widespread depression of oxidative energy delivery in all cells. Early symptoms might be nonspecific, like muscle weakness, fatigue, and poor glandular secretion.
But severe deficiency leads to
significant tissue damage and failure. The source emphasizes the characteristic 4 Ds of pellagra: Dermatitis, a typical rough, pigmented skin rash, especially in sun-exposed areas due to impaired skin repair. Diarrhea, resulting from intense irritation and inflammation of the mucous membranes throughout the GI tract, potentially leading to hemorrhage. Dementia, due to neuronal degeneration in the CNS, potentially causing psychoses or dementia. And ultimately, if untreated, death.
The GS symptoms sound particularly severe.
They can be. The inflammation can affect the entire digestive tract, leading to severe digestive issues, malabsorption, and bleeding. The source also reiterates the link between pellagra and diets heavily reliant on corn maize, which is not only low in niacin itself, but also deficient in tryptophan, the amino acid precursor, thus doubly increasing the risk.
Okay, moving on to pantothenic acid, vitamin B5.
Pantothenic acid is absolutely essential because it's a core component of coenzyme A, universally known as CoA. CoA is a central hub molecule in metabolism, primarily involved in transferring acetyl groups to carbon units.
What are its key roles?
Its roles are fundamental to energy metabolism. It's required to convert pyruvate from glucose breakdown into acetyl-CoA before it can enter the citric acid cycle. It's also essential for the breakdown of fatty acids via beta oxidation, which also produces acetyl-CoA. And it's involved in the synthesis of cholesterol, steroid hormones, and neurotransmitters.
So vital for processing both carbs and fats for energy. What about deficiency?
Interestingly, while deficiency syndromes have been clearly demonstrated in experimental animals, causing things like retarded growth, reproductive failure, graying hair, skin lesions, neurological problems, adrenal damage, the source states that no definite specific deficiency syndrome has been conclusively proven in humans under normal dietary conditions.
Why might that be?
It's likely due to a combination of factors. Pantothenic acid is very widely distributed in foods, making a purely deficient diet rare. Plus, there might be some minor synthesis by intestinal bacteria. However, its absolutely crucial metabolic role means it is undoubtedly essential for human health, even if overt deficiency disease is uncommon.
Next is pyridoxine, vitamin B6.
Pyridoxine is converted in the body to its active coenzyme form, pyridoxal phosphate, POP. POP is a workhorse coenzyme involved in a vast number of enzymatic reactions, particularly those related to amino acid and protein metabolism.
What's its most critical role?
Its most prominent role is in transamination reactions. These are reactions that transfer amino groups from one molecule to another, and they are absolutely essential for the synthesis of non-essential amino acids, the ones our body can make, for the interconversion of amino acids. PLP is also involved in decarboxylation reactions, removing carboxyl groups from amino acids important for neurotransmitter synthesis, and it seems to play a role in the transport of some amino acids across cell membranes.
What are the effects of B6 deficiency?
In experimental animals, deficiency can cause a range of problems, including dermatitis, decreased growth rate, development of fatty liver, anemia, and evidence of mental deterioration or neurological issues like seizures. In humans, dietary deficiency is relatively rare, but it can occur, particularly in infants fed deficient formulas historically. Symptoms can include seizures, dermatitis, especially around the eyes, nose, and mouth, and gastrointestinal disturbances like nausea and vomiting.
Now for vitamin B12, cobalamin.
Vitamin B12 is unique in that it contains the mineral cobalt, hence the name cobalamin. It functions as part of several coenzyme systems. The most critical function highlighted in this material relates to its role, along with folate, in the synthesis of DNA. Specifically, B12 coenzymes are essential for reducing ribonucleotides to deoxyribonucleotides, a key step in creating the building blocks needed for DNA replication.
So vital for cell division.
Exactly. This fundamental role explains why B12 is crucial for promoting overall growth and, very importantly, for the proper formation and maturation of red blood cells in the bone marrow as these cells divide rapidly.
And deficiency leads to the condition called pernicious anemia.
Yes, pernicious anemia is the classic B12 deficiency disease. It's characterized primarily by a failure of red blood cell maturation. The bone marrow produces large, fragile, immature red blood cells called macrocytes, leading to anemia. But critically, B12 deficiency also has severe neurological consequences.
What kind of neurological problems?
It causes progressive demyelination breakdown of the protective myelin sheath of the large nerve fibers, particularly in the spinal cord. This leads to neurological symptoms like numbness and tingling in the extremities, loss of peripheral sensation, muscle weakness, difficulty walking, and potentially irreversible paralysis in severe, untreated cases.
And the source emphasizes that the usual usual cause of this deficiency isn't simply not eating enough B12, right?
That's a key point. While dietary B12 deficiency can occur, especially in strict long-term vegans who don't supplement, the most common cause of pernicious anemia is actually a failure to absorb B12 from the diet. This is usually due to a lack of intrinsic factor.
That's intrinsic factor.
It's a special protein secreted by the parietal cells in the lining of the stomach. Intrinsic factor binds to dietary vitamin B12, and this complex is then absorbed way down in the terminal ileum, the last part of the small intestine. Without intrinsic factor, often due to autoimmune destruction of the parietal cells, B12 simply cannot be absorbed effectively, leading to deficiency even if dietary intake is adequate.
Okay, closely related is folic acid, vitamin B9.
Folic acid, or folate, its natural form, is another B vitamin absolutely crucial for DNA synthesis. It functions as a carrier of one carbon units, like methyl groups, that are essential for the synthesis of purines and thymine key building blocks of DNA.
So like B12, it's vital for cell replication and growth.
Exactly. It's required whenever cells are dividing rapidly. The source notes it's a potent growth promoter, perhaps even more so than B12 in some respects, and, like B12, it's also essential for the proper maturation of red blood cells in the bone marrow.
Which means its deficiency also causes a similar type of anemia.
Yes. Folic acid deficiency leads to a macrocytic anemia that looks very similar under the microscope to the pernicious anemia caused by B12 deficiency. The red blood cells are large and immature. However, an important distinction made is that while both vitamins are needed for RBC maturation, the anemia caused only by folic acid deficiency can often be corrected by giving folic acid supplements alone. But this can be dangerous because it might mask an underlying B12 deficiency, allowing the potentially irreversible neurological damage from B12 deficiency to progress untreated. That's why it's critical to determine the exact cause of macrocytic anemia.
Got it. Let's move to vitamin C, ascorbic acid.
Vitamin C's primary and most well-established function highlighted in this material is its role as a cofactor for the enzyme prolyl hydroxylase and also lysyl hydroxylase. These enzymes are responsible for adding hydroxyl OH groups to the amino acids proline and lysine after they've been incorporated into collagen precursor chains.
Why is that hydroxylation step important?
It's absolutely critical for the proper formation of strong, stable collagen fibers. Hydroxyproline and hydroxylase allow the collagen chains to cross-link effectively, forming the robust triple helix structure that gives collagen its tensile strength. Collagen is the main structural protein in connective tissues throughout the body, skin, tendons, ligaments, cartilage, bone matrix, blood vessel walls, scar tissue.
So without vitamin C, you get weak collagen, and deficiency causes scurvy.
Exactly. Scurvy is the disease resulting from prolonged, severe vitamin C deficiency. Typically takes 20-30 weeks of near-zero intake to develop fully. All the major symptoms stem directly from the failure to produce normal, strong collagen. Wounds fail to heal properly because fibroblasts can't lay down adequate collagen fibrils or intercellular cementing substance. Bone growth ceases in children because the osteoblasts can't produce the necessary collagen matrix for bone deposition. Existing bones become weak and fracture easily, and fractures fail to heal.
And effects on blood vessels.
Blood vessels, especially small capillaries, become extremely fragile because their walls rely on collagen for strength and integrity. This leads to widespread hemorrhages, tiny pinpoint bleeds under the skin, petechiae, larger bruises, purpura or ecchymoses, bleeding gums, nosebleeds, and potentially more serious internal bleeding in joints, muscles, the GI tract, or even the brain.
Other symptoms of scurvy.
The source also mentions muscle weakness and fragmentation, loosening of teeth due to degeneration of the gums and the socket holding the teeth, increased susceptibility to infections, and often fever and general malaise. It's a devastating systemic disease resulting from the failure of one fundamental structural protein.
Vitamin D is next.
The key function of vitamin D emphasized in this section is its role in promoting calcium absorption from the gastrointestinal tract. It does this primarily by stimulating the synthesis of a specific calcium-binding protein within the intestinal epithelial cells. This protein facilitates the transport of calcium from the gut lumen into the bloodstream.
So it helps get calcium into the body.
Exactly. The source mentions it also plays a role in controlling calcium deposition into bone, but its effect on intestinal calcium absorption is fundamental to maintaining overall calcium balance. Its broader roles in calcium and phosphate homeostasis are likely covered elsewhere in the source material.
Vitamin E, alpha-tocopherol.
Vitamin E's primary active form is alpha-tocopherol. The source notes that overt deficiency is rare in humans under normal circumstances. However, deficiency has marked effects in experimental animals, including infertility, hence its old name, the anti-sterility vitamin, failure of growth, and degeneration of various organs like kidneys and muscles.
What's its main proposed function?
It's primarily believed to function as a powerful lipid-soluble antioxidant. Its main role is thought to be protecting polyunsaturated fatty acids, which are components of cell membranes and organelle membranes, like mitochondria and lysosomes, from oxidative damage by free radicals. By preventing this lipid peroxidation, vitamin E helps maintain the structural integrity and function of cell membranes.
Finally, among the vitamins discussed here, vitamin K.
Vitamin K is essential for blood coagulation. Its specific role is as a required cofactor for a particular enzyme in the liver, gamma-glutamyl carboxylase. This enzyme modifies several key blood clotting proteins, specifically factors 2, which is prothrombin, 7, 9X, by adding carboxyl groups to some of their glutamate amino acid residues.
And this modification is necessary for them to work?
Absolutely essential.
Yeah.
This carboxylation step allows these clotting factors to bind calcium ions, which is necessary for them to interact properly with phospholipid surfaces, like on platelets, and participate effectively in the blood clotting cascade. Without adequate vitamin K, the liver produces inactive forms of these factors, leading to impaired blood clotting.
So deficiency leads to bleeding problems?
Yes, a deficiency results in significantly retarded blood clotting and an increased tendency to bleed. However, the source makes an important point. Primary dietary deficiency of vitamin K is actually quite uncommon in adults.
Why is that?
Because bacteria residing in our large intestine, colon, normally synthesize a significant amount of vitamin K, which can then be absorbed and utilized by the body. Deficiency typically occurs when this gut bacterial population is drastically reduced, often as a result of prolonged treatment with broad-spectrum antibiotics, rather than solely from inadequate dietary intake. Newborn infants are also at risk because their gut flora isn't fully established yet.
Wow. That is an incredible level of detail on the specific functions of each vitamin and how those functions directly explain the often dramatic and diverse symptoms of deficiency. It really hammers home the why behind these micronutrient needs.
Understanding that connection between the molecular function and the physiological consequence is absolutely key to truly grasping their importance, isn't it?
Definitely. Okay, let's turn now to the other category of essential micronutrients discussed in this material, the minerals.
Right. Minerals are inorganic elements that the body also needs, often in very small quantities, for a wide variety of structural and functional roles. Table 72 of 4 in the source gives an overview of recommended daily intakes for several key minerals, which, like vitamins, vary widely. The source also notes that the functions of major electrolytes like sodium, potassium, and chloride are covered in detail in other sections dealing with fluid balance and nerve muscle function.
Okay, so focusing on the specific minerals detailed in this section, what are the key functions and effects of magnesium mentioned?
Magnesium is highlighted primarily as a catalyst, a required cofactor for many intracellular enzymatic reactions. It's particularly important for enzymes involved in carbohydrate metabolism, like those utilizing ATP. While most magnesium is inside cells, the source notes that the concentration in the extracellular fluid, though small, has significant physiological effects.
Such as?
High extracellular magnesium levels tend to depress both nervous system activity and muscle contraction. Conversely, low extracellular magnesium leads to increased nervous system irritability, potentially causing tremors or convulsions, peripheral vasodilation, widening of blood vessels, and is particularly associated with cardiac arrhythmias, especially after events that can deplete magnesium, like a heart attack.
Calcium, we know, is hugely important for bones.
Correct. The vast majority of the body's calcium is stored in bone, primarily as calcium phosphate crystals, hydroxyapatite, providing structural rigidity and hardness. However, the small amount of calcium ions present in the extracellular fluid is under extremely tight regulation, and even small changes have profound effects on nerve and muscle excitability.
What are those effects?
High extracellular calcium levels, hypercalcemia, decrease neuronal excitability, acting as a general mental depressant, and can cause the heart muscle to contract too strongly and potentially stop in systole contraction. Conversely, low extracellular calcium levels, hypocalcemia, dramatically increase the excitability of nerve fibers, making them prone to discharging spontaneously. This leads to the muscle spasms and cramps characteristic of tetany.
Phosphorus. often discussed alongside calcium.
Yes, phosphate is the major intracellular anion, negatively charged ion. It's absolutely vital because it forms reversible bonds with countless coenzymes and compounds that are central to energy metabolism and cellular signaling. Think of ATP, adenosine triphosphate, ADP, phosphocreatine. These are the energy currency and immediate energy reserve molecules, all relying on phosphate bonds. A large amount of phosphorus is also, of course, incorporated into bone alongside calcium as calcium phosphate.
Iron is highlighted here as being absolutely essential for life.
Why? Without question. Its roles are fundamental to oxygen transport and utilization. About two-thirds of the body's iron is incorporated into hemoglobin, the protein within red blood cells responsible for carrying oxygen from the lungs to all the tissues.
And its other key role.
Iron is also a critical component of cytochromes and other related proteins, like myoglobin in muscle. The cytochromes are essential electron carriers in the mitochondrial electron transport chain, the process responsible for most cellular oxidation and the generation of ATP. So iron is required both for delivering oxygen to the cells via hemoglobin and for the cellular machinery that uses that oxygen to produce energy via the cytochromes. Life as we know it, aerobic life, would simply cease very quickly without sufficient iron.
The source then briefly touches on some important trace elements, needed in even tinier quantities.
Yes, defining trace elements as those required in minute amounts. The ones specifically discussed here are iodine, zinc, and fluorine.
Iodine's role is very specific and well known, right?
Extremely specific and critical. Iodine is absolutely essential for the synthesis of the thyroid hormones, thyroxine T4, and triiodothyronine T3, by the thyroid gland. These hormones are the primary regulators of the overall metabolic rate of virtually all cells in the body. The source notes the entire body contains only about 14 milligrams of iodine, just illustrating how potent and crucial these tiny amounts are for normal function. Iodine deficiency leads to goiter in enlarged thyroid and hypothyroidism.
Zinc is mentioned as being a component of many enzymes.
Yes, zinc serves as an essential cofactor for a large number of different enzymes involved in diverse metabolic pathways. The source gives key examples. Carbonic anhydrous, which is crucial for rapid carbon dioxide transport and elimination, found in high concentrations in red blood cells, the GI mucosa, kidneys, and various glands. Lactic dehydrogenase, important for the interconversion of pyruvic acid and lactic acid, key steps in carbohydrate metabolism, especially under anaerobic conditions. Various peptidases, enzymes essential for protein digestion in the gastrointestinal tract. So through these enzymatic roles, zinc is indirectly essential for CO2 metabolism, aspects of carbohydrate and fat metabolism, and protein digestion.
Finally, fluorine. The source says it's not actually necessary for basic metabolism.
That's the perspective presented in this material that, unlike the other minerals discussed, fluorine doesn't appear to be strictly required for life or fundamental metabolic processes. However, it does have a well-established beneficial effect in preventing dental caries, tooth decay, particularly when incorporated into tooth enamel during development.
How is it thought to work?
The proposed mechanism is that fluoride ions replace hydroxyl ions in the hydroxyapatite crystals of enamel, making the enamel harder and more resistant to acid dissolution caused by bacteria. It might also inhibit certain bacterial enzymes in dental plaque that produce acid.
But too much fluoride can be harmful, causing fluorosis.
Yes, excessive intake leads to fluorosis. Mild cases manifest as cosmetic mottling or staining of the tooth enamel. More severe, chronic excess intake can lead to skeletal fluorosis, characterized by abnormal thickening and potentially weakening of bones. The proposed mechanism for toxicity is that excessive fluoride might interfere with or inactivate essential metabolic enzymes, perhaps particularly phosphatases involved in bone and tooth mineralization, by binding to metal cofactors or altering enzyme structure. So while small amounts are beneficial for teeth, excessive intake clearly illustrates the principle that even helpful substances can become toxic at high doses.
Wow. Okay, we have covered an immense amount of ground in this deep dive. We've gone from the absolute basics of energy balance through the incredibly intricate neural and hormonal webs that regulate how and why we eat, explored the serious consequences when those systems go awry in obesity and starvation, and finally delved into the absolutely vital, diverse, and specific roles played by each vitamin and mineral, right down to the molecular level.
We really have unpacked the specific body of material piece by piece. I think it highlights so clearly the interconnectedness of all these systems. Maintaining health really depends on achieving balance at multiple levels simultaneously, from the overall calorie balance down to the precise intake of each essential micronutrient.
And the goal here, as we said at the start, wasn't just to give you a quick overview, but to really dive deep into these concepts, drawing directly from the source material to help you build that foundational understanding. Moving towards a level of mastery, really, where you can see the why behind these complex physiological processes.
Exactly. Having this kind of detailed understanding derived from the source material gives you a much more powerful lens through which to view nutrition, metabolism, health, and even disease states. You can start to see the underlying logic.
So let's leave you, our listener, with a final thought to ponder, something that weaves together the threads we've pulled from this material today. Considering the body's ancient and powerful regulatory systems, the hypothalamus, the hormones like leptin and ghrelin, the metabolic adaptations, all seemingly designed to optimize energy acquisition and storage in environments where food was often scarce and unpredictable, how does this deeply ingrained biological programming truly grapple with the unprecedented reality of the modern world, characterized by constant food abundance, easily accessible high-calorie options, and pervasive sedentary lifestyles?
Or perhaps think about it from the other end. Consider the stark specific symptoms of a micronutrient deficiency we discussed, like the fragile blood vessels and failed wound healing in sturdy. And contrast that with the tiny, precise molecular job of vitamin C, simply helping to build strong collagen molecules. How does understanding that singular, specific biochemical role instantly illuminate the fundamental cause of those seemingly disparate and devastating systemic symptoms? It reveals the underlying elegant logic of biology, doesn't it?
Keep connecting those dots.